But I'm happy to be able to share a little bit today about work on water properties. And this is what I usually don't talk about. So it's new for me. I'm just starting out with this visualization, not to put you to sleep, but to get you to think about the fact that as tides rise and fall, the tide flats dry out, and the water moves in and out of the estuary at the mouth. But you can also sort of see a bulge of blue going up onto the shore and down, and then maybe the same bulge coming back up, and a bulge of blue going out of the estuary now, and maybe the same bulge coming back in. And that's part of the message that I just wanted to leave you with today. So you've gotten it. But we're going to look in a little bit more detail at water properties. And when I say water properties, I mean this list of things, especially salinity, because it tells us about the contribution of the ocean and fresh water to the system. And then a variety of things that can be important for the organisms that live in the bay. And then a lot of things that we've been talking about in terms of food supply for bivalves, the phytoplankton and the particulates that are actually in the water. And just a few pictures here that show the variety of techniques that we have applied to this, all of which are basically things you can buy at Jack's Country Store, except maybe that the salinity meter, that long tube, which is a little bit more complicated. I wish I could get one at Jack's. All right, the take-home messages are here, and I'm just I'll step through them and come back to them at the end. But they really are they are messages about how estuaries work with data that we have collected primarily in Willapa Bay. And then ending with organisms can change water properties, but that's what we know from the rest of today's presentations, that many of these species are ecosystem engineers that influence the system. Okay, so first, an estuary is where fresh water mixes with the ocean. And as a reminder, fresh water has essentially no salt in it, and salt water in the ocean has 33 parts per thousand. So in an estuary, you expect to see that kind of gradient. And we have collected data on how much salt is in the water, all the way from the mouth of the bay to the Nassau River in the summertime. And so perhaps it is not surprising to see the trend, that is, it's saltier at the ocean and fresher near the Nassau River, but possibly a little bit surprising to see that even the Nassau River is pretty close to ocean salinity by the time you get to the Nassau Bridge. Interestingly, this line really takes a steep dive in the winter when the salinity near the Nassau Bridge can be down. And then for temperature, the bay inside really warms up in the summer, and you can see that it's really in that region below the fattening line that Brett was just describing, where there are warmer temperatures. So the ocean in the summertime is colder than the water running off the land, and that generates this temperature gradient. We can also see gradients of the material that organisms are using to build their bodies. So on the left, I have a picture up there that will be for eelgrass, and on the right, a picture that will be for oysters, and they're set up in both cases to be close to the ocean on the left-hand side and near a river on the right-hand side. And then that vertical axis, which says delta 13C, that's carbon in parts per thousand, is an isotopic tracer. It's a chemical tracer that tells what an organism is actually eating. So if you are a vegetarian and you are a carnivore, and I take a piece of your hair and I look at some of the isotopic signatures in your hair, I can actually tell what your diet is based on that. And that's pretty cool. So it's a really powerful technique to be able to use. You might think, oh, oysters are eating the same thing everywhere, or eelgrass, it's photosynthesizing the same way everywhere. Why would it have a different chemical signal? But in fact, the carbon sources that come from the land and that come from the ocean have really different intrinsic signals. So I just put that up as a little code on the left-hand side. So higher values on these axes mean, yep, eating more stuff from the ocean. Lower values mean using more stuff from land. And in fact, we should see really dramatic gradients in both the filter feeders that are eating the phytoplankton that have carbon sources that differ from where they come from, and also in the eelgrass, which is just using carbon sources from different places. So this means that if you were eating eelgrass from the mouth of the bay or eating eelgrass from close to the nasal bridge and I took a piece of your hair, I could tell which kind of eelgrass you were eating. There you go. All right, so one more thing that differs from the ocean up into the estuary is something about the sediment type. So again, ocean on the left, river on the right, and the vertical axis, organic content, basically tells you about the muckiness of the water. So pretty sandy, or sorry, the muckiness of the sediment, pretty sandy on the low end, but getting up towards cake frosting as you get up into 9 and 10% organic. And this sediment condition is another dramatic gradient across the estuary. There are, of course, lots of places where there's heterogeneity within parts of the estuary, but this is the large-scale pattern of cake frosting near the rivers and sandier sediments. This also can affect what goes back up into the water. So here, this is a picture of how many suspended particles are actually in the water. And here, I'm looking at shallow water and deeper water just because of this idea of resuspension being a lot easier if the water is very shallow. It's easier for small waves to pick things up off the bottom. And this is the messiest graph that I'll show you, but I will also say that there's a statistically significant relationship here in shallower water. That's where the fine particles get kicked up. And that also there are capacities for our ecosystem engineers, eelgrass and oysters, to actually augment the amount of particles that are in the water based on our data. Okay, so that is from the ocean up to the river, and that just tells you about the gradients that are experienced in estuaries and that are evident in Willapa Bay as well. But there's another part of this gradient which has to do with how temporally variable, how much variability there is over time in the water properties that an organism experiences at a particular place. And I'm really going to emphasize two aspects of that temporal variability. So at the ocean, as Brett just mentioned, in the summertime especially, northwest winds blow. We have beautiful bright sunshine, but those northwest winds actually cause water to be forced offshore and a vacuum close to shore that brings deep, cold, nutrient-rich, high carbon dioxide, low pH water to the surface right outside the bays. So that is a process that has about a week time interval. It's nice for a week, and then it's really rainy and yucky for a week, even in the summer, and then maybe it's nice again for a week. In contrast, the major forcing factor at the river end of the estuary for organisms that are fixed in place is how much of that river water are they actually getting at any particular time. And that's a function of what time in the tide it is. So tidal action is really an important part of what drives the variability, the scale of the variability near these rivers. And I'm going to focus here on pH because this was a part of a project that we were doing to try to understand the pH conditions that oysters were experiencing at different parts of the bay. Okay, so near the ocean, long time variability, mostly from upwelling, that's the idea. And near the river, short time variability, mostly from water level going up and down associated with the tides. So overall, as we drove our boat around and measured the water properties, we actually were measuring the water properties when there was a huge phytoplankton bloom near the mouth of the bay. It was coming in from the coastal ocean. The carbon dioxide in the water was actually less than you would expect from the atmosphere. The photosynthetic organisms had used up that carbon dioxide in their photosynthesis and taken it out of equilibrium with the atmosphere. By the time we got up into those rivers, we were getting that lower pH water coming in from the rivers, and also a lot of material coming in from the rivers, which was then decomposing and releasing carbon dioxide. Okay, so that's the big spatial pattern. And then I'm just going to take you through 3 different places. So first, close to the mouth, quite a lot of variability in pH in 2 summers when we measured it. And that variability was happening at the scale of about a week. And every time there was a big upwelling event, we got a low pH event. So upwelling is actually an inverse scale on the right-hand side. Those dots that are the farthest down are actually the most upwelling, but that's to be able to see that those high upwelling times are associated with the lowest pH. Farther into the bay, that correlation completely disappeared. In the middle of the bay, we actually got pretty relatively stable pH readings as we were recording through the summers. And when we actually looked at that as a function of time of day, we saw a really strong biological signal. During the night, from midnight until 9 a.m. or so, was dark. Everything in the water was respiring and releasing carbon dioxide, and that reduced the pH in the water. During the day, as soon as it got light up until sunset at the end of the day, the net process was photosynthesis from phytoplankton, eelgrass, all the things that are green in the bay. They use up carbon dioxide and actually increase the pH. And that daily variability is actually about as much variation as we've seen since the Industrial Revolution in the ocean as a whole. So biology really matters. Okay, and then as we get up to the river part, what you see is this really strong variation at very short time scales in the pH variability. So on the order of 0.5 pH variability, pH units on a tidal basis, with the most extreme pH variability being at those times when the daily tidal exchange was the greatest. When it went from the highest high tide to the lowest low tide is the times when the pH also varied the most, associated with that low pH water being able to extend further downstream on a low tide. Okay, well, I have really just barreled you with a whole bunch of information about variability. I'll just summarize by saying this was really to just talk about Willapa Bay as an estuary that is quite different in different places and also at different time scales. We know this from first principles because it is an estuary. We can also measure it with salinity, temperature, pH, and sediment type, and we have these biorecorders in eelgrass and oysters and other fixed organisms that can help us understand the energy sources that are supporting different parts of the bay. So what? Well, I'm going to come back to this oceanic plankton bloom being the major source of food, but that would be one important source of food, especially at the north end of the bay. And then this whole set of conditions that organisms are exposed to keep in mind in planning purposes that the bay is not a homogeneous entity. All right, on to residence time. And I'm just going to say that this is sort of like tidal meteorological forcing, which I wrote down on my hand to be able to remember, but that's what Tomas told us about as a key component of how fast a water body turns over, so we can understand how much effect an organism is going to be able to have in drawing down the food supply that's available there. Okay, so these pictures in the light blue show the spatial extent of water in the bays at low tide. And this means, as I think we have heard, but it's also just good to internalize, half of these bays dry out at low water and half of the water leaves them on low tide. And that's a huge, huge tidal exchange, or huge turnover. That is a very strong amount of tidal meteorological flushing. And that has led to estimates of extremely short residence times on the order of 0 to 1 days, because there are 2 tides a day and half the water is turned over every single time. That is a gross oversimplification, because if this were the case, it would be really impossible for any organism ever to make a difference at all for the water in these bays. So the physicists who have turned biologists have become more complex within Willapa Bay as well, and given us modeling results like this, where they take into consideration not just the volume and the tidal exchange, but the shape of the bay. And have documented or predicted that water takes about a week to turn over in the north end of the bay. And in the summertime, when there's very low river flow, so this is actually a model run that assumes no river flow whatsoever. Also a gross oversimplification, but sometimes you have to start there. That water can be resident in the south part of the bay for over a month. And that has strong implications for the ability of organisms to be able to influence that water, and also for how long, how long something that comes in from the mouth would actually stay in the bay. Okay, but it gets even more fun. Most recently, Neil Banas, who is our collaborator oceanographer and did a lot of work here during his thesis and his postdoc, looked at how long water stays on the tide flat. And you might think, I know that, it only stays there until it's low tide, and then it's gone from the tide flat. But in fact, if you think back to the first picture that I gave you of the water going down, coming off of the tide flat, but sort of going down into a reservoir and then feeding right back up onto the tide flat, that's what shows up in these models. So the water drains off every day, but in fact, the same water can sit on a tide flat for up to 3 days on average, because the same water that drains off also turns around and goes right back on to the tide flat. So this to me was a really weird thing to have to come to terms with, but actually we have empirical data that is really consistent with that model as well, of the tide flats being rebathed by the same old water that just used to be there. Okay, so for residence time, we know about residence time because of modeling, and the increasingly sophisticated models give us an increasingly sophisticated insight into how long water stays in a place and where it goes. We do have both biological and physical data that the models are actually pretty good in this case, so that's exciting. And then why do we care? Again, it goes back to this issue of bivalve carrying capacity, but it also determines things like how warm water can get, and also the dispersion of larvae. For instance, where will larvae go if they're produced in one place? How long will they stick around? Okay, and then finishing up. I'm just looking for my time. Good? All right, good. I've been talking fast, apparently. Okay, so finishing up, where we return back to this issue of ecosystem engineers and what organisms can do to their environment. One thing just to keep in mind is that this issue of residence time fits in with ecosystem engineers because a patch of eelgrass, for instance, that can draw down carbon dioxide and release oxygen in the daytime will have more influence on a patch of water that spends a long time in eelgrass than one that spends a short time in eelgrass. And similarly, water that sits for a month in the south end of Willapa Bay might be more accessible for bivalves to filter it out than one that sits for a week up at the north end of Willapa Bay. Okay, we do know from empirical measurements that organisms do have strong influences on the water that flows across them or flows through them. Eelgrass can slow down the water. It can reduce the turbulence of the water. It can even it out. It protects shoreline. We do know that oysters can reduce the amount of phytoplankton in the water, although 2 publications out of my lab actually show 2 different results on that. So that's ecology for you. So there are probably, we need to know more about the conditions under which oysters can reduce the chlorophyll in the water. And the so what part is, yeah, we're back to ecosystem engineers. So I will actually just spend a little bit of time on this, which was an effort on my part to think about what do we know about different habitat comparisons. I put mudflat on one side and eelgrass on the other side, and a number of additional habitat types in our estuaries in the middle, and then drew lines between pairs of habitats that I thought would be most interesting to compare. It's not all of them. I didn't draw lines everywhere, but I did draw quite a few lines. So there's 13 different pairs of habitats. And then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different response variables of what might be happening in those habitat types. There on the right-hand side is the bird response, and in terms of comparing birds in pairs of habitats, we, as far as I know, have no data, although the Wildlife Refuge is working on a project now that is beginning to accumulate some of those data. We have quite a lot of data in the mud and eelgrass comparison for nekton, which is part of what Beth talked about earlier. And the blue sections are work on nekton responses in Puget Sound, so she also talked about that earlier. And we have some information about how eelgrass does in comparisons in some of the oyster aquaculture conditions, and that went into the review that Beth talked about earlier. What I've really been talking about are the first two columns of what are water and sediment properties in these estuaries, but in fact, when we look at the water and sediment properties that we've studied in different habitat types, it's a very few studies that have been done. So, but not all gaps need to be filled. But in some cases, it may actually be really helpful to spend a little bit more effort really understanding what these organisms are doing and how other organisms care. Okay, so this then also gets to what are these organisms doing. This modeling effort from Neil Banas looked at an assumption of all the phytoplankton being based in the ocean and tides bringing phytoplankton in. And he documented that even though 85% of his modeled phytoplankton would just go right back out of the mouth of the bay, that it was actually those bivalves in the north end of the bay that were stunting the growth and reducing the food for the bivalves in the south end of the bay. And I am a co-author on this, so I sort of agree with this, but on the other hand, I'm also an empiricist, boots on the ground ecologist. And so you might think at that point, well, if we could feed those oysters at the south end of the bay a little bit more, they should grow better, right? So we did that experiment. We have used this tile technique where we put small oysters onto tiles and then we can measure their growth over time. We know that in the summertime, oysters are putting on about 12 millimeters of shell per month at the north end of the bay and about 8 millimeters of shell per month at the south end of the bay. That's the stunting. And then we did an experiment twice, actually, where we had a scow anchored out there, or scow or fluxy anchored out there. We grew oysters on tiles. We added phytoplankton food to some of them. We literally dripped food into their mouths, feed them more. And we thought, okay, we're gonna find out if they're food limited and we can fix it by feeding them. And of course it didn't work that way. Food didn't matter at all. All the oysters grew great. And this means that it might not just be food, but there may also be issues associated with sediment type that could be really important that for where oysters, for the productivity of oysters. It is often the case, but not always the case, that oysters grow faster off bottom than on bottom. This is, to me, just a really good indication of how important it is to take multiple approaches to trying to answer a question, and also a good realization that we are not to the bottom of what actually influences oyster growth. Okay, so take-home messages again, and then maybe a different way of take-home messages with some questions. Some things that came up to me about gaps in what we still know about what's in the water.